The document that I'm going to be scribbling in the video would be provided through a link in the description, and also the link would be there in the first comment. Please do check it out, and let's get on with the video. Alright, so now let's take some conditions and understand the regulation of the lac operon. Now, the metabolism of lactose is only necessary when lactose is present in the medium, and it is and the bacteria needs to use the lactose as a carbon source. So when lactose is absent, so if lactose is absent, then the metabolism of lactose is not required, and all the genes that are present, that is all of these genes that are present, they can be switched off, and that is exactly what happens. When lactose is absent, what happens? If I draw it over here once more, if this is the Z, this is the Y, and this is the A, this is the promoter and this is the operator, and this is lac I, and this is the promoter for lac I, then if lactose is absent, the repressor molecule, it will be formed, and this will bind to the operator. This will bind to the operator over here. It will bind to the operator, and it won't allow the transcription of the structural genes. That is, in other words, once the operator is bound to by the repressor molecule, that is lac I, so let me write it down, lac I, then the RNA polymerase cannot bind to the promoter, and hence the transcription is not done. So over here the expression is absent. So this is the case when lactose is absent, right? When lactose is absent. Now let's say when lactose is present, when lactose is present, what will happen? The lac Y that was being secreted at a basal level would bring in lactose. Let's say this is the membrane, and over here the basally secreted lac I brings in some lactose, and what will happen? This lactose will get converted into something which is called allolactose. It is going to isomerize to allolactose. Now this allolactose, what it will do, it will bind to the repressor that is already bound to the operator. It will bind to the repressor, and it will change the conformation of the repressor. So let me write it down. Allolactose, it will bind to repressor, change the conformation of the repressor, and because of this change in conformation, what will happen? The repressor that was previously bound to the operator will fall off. Repressor will fall off, and once the repressor falls off, the promoter is free for the RNA polymerase to bind, and there won't be any obstacle for the RNA polymerase to bind to the promoter right now, and hence the expression of the structural genes, that is ZYA, that will occur. So this is what is happening when lactose is present. So these two are the situation. When lactose is absent, the lac I, it will bind to the operator of the lac operon and it will inhibit the synthesis of the structural genes. Whereas when lactose is present, now when lactose is present, basically you need more of these structural genes to be active, right? More of these structural genes to be active, because now the bacteria would want to utilize lactose and do its metabolism. So when lactose is present, to break it down, it needs the lac Z. To bring the lactose inside, it needs the lac Y, and to transfer the acetyl group, it needs the lac A. So that is why it will synthesize more of it will synthesize more of the structural genes so that it can metabolize lactose properly.